Hello everyone, I'm Anas and this is lecture 24 or session 24 within chapter 24 of the Advanced Medical Science Liaison and Medical Monitor Certification course program by CCRPS. The chapter is about conceptual and theoretical monitoring, while the topic for the session is about monitoring of drug confirmation and drug authentication interaction trials. Introduction. The safety and success of pharmaceutical therapies rely on meticulous drug confirmation trials and drug authentication trials. These phases are pivotal to clinical research and drug development, ensuring that drugs perform as intended and are verifiable in their origin, composition, and identity. Key points. Drug confirmation. Definition for the drug confirmation. Interaction trial. Drug confirmation ensures that a pharmaceutical product performs as intended by validating its pharmacodynamic and pharmacokinetic properties, PD and PK properties. The key elements in drug confirmation interaction trial. On target efficacy, the trial confirms whether the drug achieves its mechanism of action at the desired site. An example here, cetuximab confirmation ensures specific binding to EGFR in oncology trials. Dose-response relationships validating that increased doses yield proportional therapeutic responses within a safe range, as for example, beta blockers demonstrate dose-dependent reductions in heart rate during hypertension studies, confirming therapeutic windows. The importance here, proper drug confirmation avoids false positives, ensuring therapeutic claims are backed by robust evidence. And the challenge here, variability in patient responses due to genetic age or demographic factors that may obscure confirmation data. Another challenge, complex multi-target drugs requiring extensive pharmacodynamic PD analysis. The outcome here by validating effects, confirmation trials pave the way for advancing drugs to larger clinical phases or approvals. While, on the other hand, the drug authentication interaction trial or trials, here the definition as drug authentication ensures that the administered drug is genuine, composed of the correct active ingredients and free of impurities. It encompasses identity verification, purity testing, and traceability. And here's a summarized diagram on drug, summarizing the drug authentication process as from left to right, identity verification, purity testing, and traceability. The key strategies in drug authentication, identity verification, ensure the chemical structure matches manufacturing standards, as for example, authenticating biosimilar monoclonal antibodies to match originator biologics in rheumatology trials. Purity testing detect the presence of impurities, contaminants, or undeclared additives that can compromise patient safety, as for example, chromatographic methods like HPLC ensure chemotherapy formulations like paclitaxel meet purity standards. Traceability monitor the drug's production and distribution path to detect issues like unauthorized sources or counterfeit drugs. As for example, here RFID tagging confirms authorized sources during vaccine supply chain audits. The challenges here are counterfeit drug infiltrating supply chains, especially in low regulation regions, potentially threatening trial validity and patient safety. The outcome here, authentication, guarantees that the drug administered meets regulatory, ethical, and manufacturing uh, standards. Monitoring strategies, confirmation-specific techniques. We have pharmacodynamic PD validation, use biomarkers like IL-6 to confirm intended therapeutic impacts, as for example, monoclonal antibody effectiveness. As for example, reduction of hemoglobin A1C in a diabetes trial validates insulin analog efficacy. And here is a summarized diagram on ensuring drug safety and trial integrity, as we have confirmation-specific techniques, pharmacodynamic validation, pharmacokinetic profiling, patch and lot verification, chemical fingerprinting, authentication-specific techniques, all towards preventing safety risks and ensuring trial integrity. Pharmacokinetic PK profiling confirmed that drug levels in plasma align with predicted models, as for example, monitoring anti-epileptic drug levels ensures steady therapeutic doses or dosing. Authentication-specific techniques, chemical fingerprinting, methods like spectrometry identify the molecular fingerprints of drugs, as for example, identifying contamination in parenteral drugs via mass spectrometry. Batch and lot verification match drugs to their manufacturing records to ensure traceabilities, for example, linking vaccine doses with chain of custody records during clinical studies. The outcome here, these targeted strategies prevent safety risks and ensure trial integrity. Trial design considerations, endpoint selection, choose clear metrics for both confirmation and authentication. Example, confirming tumor size reduction, confirmation versus validating chemical stability of the drug, authentication. Blinding and randomization ensure authenticated drugs are used consistently across control and active groups to minimize bias. Challenges here: difficulty coordinating validation endpoints with authentication objectives during complex trial designs. The outcome here: design considerations help reduce trial risks and improve reliability um, of outcomes. And here is a summarized diagram on streamlining clinical trial design, as from left to right, endpoint selection, binding and randomization, addressing challenges. Challenges in monitoring. As first, here we have a summarized diagram on overcoming drug monitoring challenges. As confirmation challenges includes efficacy evaluations, patient variability, and authentication challenges includes decentralized supply chains and counterfeit risks, all toward challenges in drug monitoring. So we'll start with the confirmation challenges. Patient variability complicates efficacy evaluations, as for example, chronic pain drug responses differ significantly across age groups. Authentication challenges, counterfeit risks persist, especially in decentralized supply chains, as for example, tackling falsified anti-malarial drugs in low regulation areas. The outcome here: combating challenges requires advanced monitoring technologies, stringent oversight, and skilled physician medical monitors, or MMs. Safety and ethical considerations, data integrity risks. In data integrity risks, secure databases and thorough auditing ensure raw confirmation and authentication data are tamper-proof. As for example, producing tamper-proof lab records during insulin efficacy trials. Fair access to genuine drugs, authentication safeguards patient populations against counterfeit or falsified drugs. As for example, WHO's pre-qualification programs ensure testing drugs like anti-tuberculosis medications meet global standards. The outcome here: these safeguards reinforce ethical practices and patient safety. Next, we have tables to summarize what we have covered through this lecture. We have table on key differences between drug confirmation and drug authentication interaction trials. For each aspect here, there is a comparison between both. As for instance, for definition, drug confirmation interaction validates the drug's intended therapeutic effects, while for drug authentication interaction, verifies the drug's identity, composition, and origin. Other aspects to compare in between as main goal, focus, methodology, and example. Key on its table on key components of monitoring. Component focus example, components we have on target efficacy, identity verification, purity assessment, dose response accuracy, traceability. A table on tools and techniques for drug confirmation and authentication tool technique purpose example. Tools or techniques, we have pharmacodynamic, PD validation tools, chemical fingerprinting, pharmacokinetic profiling, PK profiling, batch lot analysis, portable authentication devices. A table on challenges in drug confirmation and authentication monitoring. Challenge type example. Challenges, we have interpatient variability, counterfeit threats, endpoint standardization, data integrity risks, supply chain complexity. Table on examples of real-world applications. Trial scenario, purpose, outcome. The trials or scenarios, we have cancer therapy trials, biosimilar verification, chemotherapy drug purity, vaccine traceability, chronic disease therapy. Application table scenario. For each scenario, we have key principle applied, key actions, timeline, and what to watch out for. As scenarios, we have confirming on target binding efficacy of cetuximab in cancer therapy trials, verifying batch authenticity and purity of paclitaxel in chemotherapy trials, ensuring dose response effects through beta blocker validation in hypertension studies, tracking biosimilar equivalence to originator monoclonal antibodies, measuring IL-6 reductions for cancer drug validation endpoints, cross-checking participant reported glucose data during insulin analog confirmation trials. More in scenarios using RFID tagging to ensure traceability of drug patches in international clinical studies, validating plasma levels of anti-epileptic drugs through pharmacokinetic PK profiling, authenticating the purity of intravenous chemotherapy solutions during clinical trials, and we have monitoring dose-dependent safety outcomes for new anticoagulant validation studies. Next, we will revise what we have covered through this lecture through some multiple choice questions or MCQs. Question 1, what is the primary goal of drug confirmation trials? A, to determine the origin of the drug ingredients. B, to validate pharmacodynamic and pharmacokinetic PD and PK of the drug. C, to ensure the drug is sourced ethically, or D, to identify counterfeit drugs in supply chains. So definitely it's B, as the primary goal of drug confirmation trial is to validate pharmacodynamic PD and pharmacokinetic PK of the drug. As drug confirmation trials are focused on confirming that the drug performs its independent therapeutic functions by assessing PD, pharmacodynamic effects, and PK pharmacokinetics, absorption, distribution, metabolism, and excretion, ADME, as for the steps or processes of PK. Question 2 of PK. Question 2, which of the following is critical to drug authentication trials? Is it A, monitoring dose response relationships, B, verifying the drug's composition and purity to ensure it is genuine, C, validating long-term efficacy metrics, or D, assessing biomarkers for disease progression rates? So definitely uh, uh, the what is critical for drug authentication trial particularly is B, verifying the drug's composition and purity to ensure it is genuine, as B is the answer. As drug authentication trials focus on verifying the authenticity of the drug, ensuring it contains the correct active ingredients, is free of contaminants, and matches manufacturing protocols. Question 3, why is high performance liquid chromatography, HPLC, widely used in drug authentication trials? Because A, it tracks patient-reported outcomes, B, it separates and measures active ingredients purity in drug formulations, C, it tests long-term clinical efficacy in various populations, or D, it establishes dose-response relationships for therapeutic windows. So definitely it's B, as the HPLC, widely used in drug authentication trial because it separates and measures active ingredients purity in drug formulations. as HPLC is a chemical fingerprinting technique used to analyze and ensure the purity of drugs by separating and quantifying their components in clinical trials. Question 4. Which challenge is most often associated with drug authentication trials? Is it A, response variability in immune biomarkers, B, counterfeit drugs entering the supply chain, C, adherence discrepancies in dose schedules, or D, exceeding therapeutic window um, efficacy? So definitely it's B, counterfeit drugs entering the supply chain, as B is the answer, as is the challenge that is most often associated with drug authentication trials, counterfeit drugs entering the supply chain. As one of the key challenges of drug authentication trials is combating counterfeit drugs, which infiltrate supply chains and require advanced methods like RFID tags or spectroscopic tools for detection. Question 5. How do batch and lot verification techniques ensure data integrity in drug authentication trials? A. They trace manufacturing consistency to ensure originality of drugs. B. They validate pharmacokinetic PK blood levels across demographics. C. They track heart rate reductions during dose response tests, or D, they analyze cell level biomarker responses. So the answer is A, how do batch and lot verification techniques ensure data integrity in drug authentication trials? As in option A, they trace manufacturing consistency to ensure originality of the drugs. As batch and lot verification confirm that the drugs used in trials are ethically sourced and consistent with manufacturing standards, ensuring traceability and compliance throughout distribution. And here we come to the end of this lecture. Thank you very much and goodbye.